Today I will be going over the different processes of muscle contraction, more specifically, innervation of muscle fibers at the neuromuscular junction, excitation contraction coupling, and the cross-bridge cycle. In this section, I will be going over the events at the neuromuscular junction. In this top illustration, we have a nerve impulse innervating a muscle fiber at the neuromuscular junction. An action potential arises from the motor neuron onto the axon terminal at the neuromuscular junction. The action potential causes voltage-gated calcium channels to open, and calcium enters the axon terminal, causing the neurotransmitter acetylcholine to be released. Acetylcholine binds to the receptors in the sarcolemma, opening ion channels in the receptors that allow an influx of sodium into the muscle as it moves down its concentration gradient, and potassium out of the muscle. This exchange of ions moving in and out of the cell ultimately disrupts the resting membrane potential and causes the generation and propagation of an action potential across the sarcolemma. In this section, I will go over the steps in the excitation-contraction coupling. In this illustration, we have an action potential represented by the red arrows being propagated along the sarcolemma and down the T-tubules. The transmission of the action potential along the T-tubules causes the T-tubule proteins to change shape, allowing calcium to be released from the lateral sacs of the sarcoplasmic reticulum. The free-flowing calcium shown in this illustration then binds to troponin, which removes the blocking action of tropomyosin, ultimately exposing the myosin binding sites on actin. The binding of myosin to actin then forms cross bridges to begin the sliding of the filaments, also known as muscle contraction. This section will consist of the series of events responsible for the cross-bridge cycle. The cross-bridge cycle, or sliding filament theory, consists of 4 stages represented by these figures. Figure A represents the cross-bridge formation. Figure B shows the power stroke. Figure C shows the cross-bridge detachment. And finally, Figure D shows the cocking of the myosin head. Cross-bridge formation occurs when energized myosin heads attaches to an actin myofilament, forming a cross-bridge. This stage requires ADP and inorganic phosphate to be present. Stage 2 represents the power stroke, where ADP and inorganic phosphate are released and the myosin head cocks back, pulling the actin filament towards the M line. Stage 3 represents the cross-bridge detachment. In the presence of ATP, the link between the myosin and the actin weakens, and the myosin head detaches as a result. At this point, the cross-bridge breaks. Stage 4 represents the cocking of the myosin head. As ADP is hydrolyzed to ADP and inorganic phosphate, the myosin head returns to its pre-stroke high energy or cocked position.